All right, we're going to do reproductive endocrinology. This is going to be basically a four-part lesson. The fourth part's going to be on male reproductive endocrinology. The first three parts on female reproduction. My sources for this, primarily Williams Gynecology, 2nd edition, chapter 15, Costanza's Physiology, chapter 10, and Wikipedia. Real quickly, I'm going to go over some definitions and terms to make sure that everyone's on the same page, including me. So, for example, we have these terms endocrine, paracrine, autocrine, and what do they mean? It's basically different types of cell-to-cell communication. There are more types than this, for example, gap junction communication and so forth. But this specific cell-to-cell communication, so with endocrine, what we're talking about is a cell in one part of the body sending out a signal to a cell in another part of the body by secreting some type of hormone that will travel through the bloodstream and act at a distant part of the body. Now that's pretty easy. However, paracrine, autocrine, the definition is going to be slightly different depending on which textbook you use. For example, autocrine, when in one textbook, in most textbooks actually, autocrine is defined as any hormone or any chemical that is secreted from the cell and turns back and acts only on that specific cell. But here's the problem. We have this cell. It's secreting some substance. Some of that's going to be acting on the same cell, but some of the substance that's secreted is going to act on its neighboring cells that are the exact same kind of cells. So, for example, the exact same kind of tissue, the exact same kind of cell within that tissue can also be acted on by that same chemical. So is that autocrine or is that paracrine? It's not endocrine because it wasn't secreted into the blood and acting on a distant tissue, but it was secreted from the cell. It acted on the very same cell, but it also acted on the neighboring cell. Most textbooks would argue that this would be paracrine and autocrine at the same time. However, in some textbooks that I've read, this would just be autocrine. So autocrine, the way I'm going to use it, is anything that's secreted from a cell and acts on the same cell type locally. Where paracrine is any substance that's secreted from the cell and acts on a different cell type that is local. It doesn't get secreted in the blood, it gets secreted locally, it acts on a different cell type. That I will call paracrine. All books will call that paracrine. Some books will also call this type of thing paracrine, but I will call it autocrine. Another set of terms that can be confused are neurotransmitter and neurohormone. A neurohormone is the same thing as a neuroendocrine secretion. I'll probably just use the term neurohormone. With a neurotransmitter, it's going to be, here's your cell body of your neuron. It's going to have an axon. It's going to have a synaptic area. And at that synaptic area, there's going to be another cell, and it's going to secrete things from the axon terminal and act directly on the cell. Whereas in a neurohormone, you're going to have your neuron cell body, you're going to have your axon. At the end of the axon, you're going to have bloodstream, and it's going to secrete stuff into the bloodstream. So that's a neurohormone or a neuroendocrine secretion, and it's the same concept as just any other hormone, only it's secreted by nerves instead of some other tissue. The main example that we're going to use of neurohormone would be gonadotropin releasing hormone, and then a good example of a neurotransmitter that's important in reproductive endocrinology would be dopamine. Dopamine and gonadotropin releasing hormone. There are two main categories of hormones. You have your steroid hormones, you have your peptide hormones. For the most part, we'll talk about a few exceptions to this rule, but for the most part, the receptors for steroid hormones are going to be intracellular, while peptides are going to be on the cell surface. Steroids are going to be nonpolar, and so they can diffuse into a cell and bind with a receptor inside the cell, whereas peptides are not going to be able to diffuse into the cell, so they have to bind with the receptor on the cell surface. And again, I'll get to the exceptions to these rules later on. When hormones are released in the blood, they're released in very, very low amounts. And so one thing that's really important concept is that the hormone receptors, these specific receptors have to have high affinity, and high specificity for their respective hormone. And so if there's just a little bit in the blood, the receptor has to be able to nab that one up, that specific hormone. It has to be able to nab it and bind it as tight as possible in order for them to have any effect. So now let's talk about a few of the peptide hormones. The important peptide hormones in reproductive endocrinology are luteinizing hormone, follicle-stimulating hormone, thyroid-stimulating hormone, human chorionic gonadotropin hormone. Now, all four of these hormones are members of a family of glycoprotein hormones. And why is that? Because they all share the same alpha subunit. So this alpha subunit is a glycoprotein that binds with one of the four beta subunits to form an active dimer. Within the blood, you can find the free beta subunit floating around, but it has no physiological activity until it's bound to the alpha subunit. This is important because these hormones can have some overlap in their receptor binding. For example, HCG, or human chorionic gonadotropin, and luteinizing hormone both have the exact same receptor. We're going to talk about later on what all the receptors are, but it's the LHCG receptor, and because of that, they will both have the same activity. And again, HCG and TSH can have some overlapping binding of the TSH receptor. So if you have high levels of HCG in your blood, it can cause hyperthyroidism. So we can get hyperthyroidism from HCG. Now, when you're doing a pregnancy test, what are you checking for? You check for beta HCG, or this HCG beta subunit. Why not check for the whole thing? Because only the HCG subunit it's produced by during pregnancy by the syncytia trophoblast, and with very, very rare exceptions, that's about the only place it's produced in a significant amount. So LH and HCG, specifically the LH beta and the HCG beta, they have 80% homology within their gene structure. They come from the same gene cluster. They're in different genes. They're in the same gene cluster, and they have 80% homology. And so the difference would be about 24 different amino acids added to the end of the HCG beta protein. And because there's a difference, a 20% difference in these two hormones, we can have very specific assays to check for either LH or HCG and differentiate them. A few more peptides that are clinically important in reproductive endocrinology would be the inhibins, which you have Inhibin-A and Inhibin-B, Activin, and Follistatin. Now, Inhibin-A and Inhibin-B, they both have the same alpha subunit. So it's important to note that this is different than the alpha subunit that's for FSH, LH, HCG, and TSH. It's a different alpha subunit, but it has an alpha subunit and a beta subunit. You can either have the beta A subunit or the beta B subunit. So this comes together as a homodimer, so you have alpha beta A or alpha beta B, and that will produce Inhibin-A or Inhibin-B respectively. Now, Activin it has no alpha subunit. It is only the beta subunit. So it's the identical beta subunit of Inhibin-A or Inhibin-B, and you can either have one of two different homodimers or a heterodimer. So you can have beta A homodimer, beta B homodimer, or beta A beta B heterodimer. And then follistatin is structurally unrelated to either of these. So how they get their name? They got their name by testing their action on follicle-stimulating hormone production. So inhibin, just as its name suggests, inhibits FSH. And activin will increase FSH. While follistatin, it's probably active by preventing activin binding. So it's not exactly known how follistatin works, but it's probably through binding the activin receptor and preventing activin from binding to it. Recently, there have been a number of additional beta subunits that have been identified. Their exact importance is relatively unknown. And it's also important to note that inhibin is probably the most important of these three as far as taking care of gonadotropins. So inhibin is known to inhibit FSH. It's probably better at inhibiting FSH than activin is at activating FSH. So inhibin most important in the gonadotropins. However, these peptides have also been shown to act on LH and GnRH, so luteinizing hormone and gonadotropin releasing hormone. However, not nearly as well as its effect. It's not nearly as strong as its effect on FSH. And the names, again, these names are derived from how they affect FSH, but the names can be a misnomer. For example, activin, even though it has activating effects on FSH, it can also negatively influence, so it can negatively influence germ cell survival, so germ cell survival in the developing ovaries, and it also has a negative effect on germ cell activation during folliculogenesis. Okay, next we have the steroids. Specifically, these are the sex steroids. But the steroids they're all synthesized from cholesterol. So up here we have a molecule of cholesterol, and you can see that this has 27 carbons. And all of the steroids they're classified by the number of carbons that they have. So, also all of the rings are usually lettered. So this would be ring A, B, C, and D. And so when they're talking about, whenever different texts and stuff will talk about actions on synthesis of the different steroids, they may refer to the rings or they may refer to the carbon number. And so just a quick overview of these, the progestins, glucocorticoids, and mineralocorticoids all have 21 carbons. These are the 21 carbon series. All androgens have 19 and all estrogens have 18. So by keeping this in mind, you can come back up here and you can look at the structure of cholesterol, and you can see that here we have 21 carbons. If we were to cut all of this off, we would have 19 carbons, and then if we cut this one off, we would have 18. Before we talk about the exact pathway that produces these steroids, first I want to talk about the enzymes. So cytochrome P450 is the enzyme. It's a superfamily of enzymes that's important in drug detoxification as well as steroid synthesis and just basically keeping your bloodstream clean and producing the hormones that you need. Usually cytochrome P450, you'll see it abbreviated as P450 or CYP. So CYP stands for cytochrome P450, and usually if you see CYP, it'll give you like a reference at the bottom, like a specific CYP, so CYP-C11, for example. Now, out of these P450s, they're located, so I have listed in here the location. For steroid synthesis, most of it takes place inside of the endoplasmic reticulum, specifically the smooth endoplasmic reticulum. However, most of the time when talking about the synthesis of steroids, it's going to list the exact reaction as the enzyme. So 11-hydroxylase, 18-hydroxylase, 17-20-lyase. These are going to be listed as the enzymes rather than listing the really large P450 enzyme name, this is just basically truncated names as well. And I could be wrong about this, but I think this nomenclature originally started because these reactions were detected, but the exact enzyme that was performing it was unknown, and so later on they discovered, so they named these enzymes based on their reactivity, and then later it was discovered that these were P450 proteins. The reason they're named P450 is because whenever you have them in a reduced state with carbon monoxide attached, they will absorb light at 450 nanometers. So that's why they're called cytochrome P450s. Now, steroid synthesis happens in basically 4 steps. So we're going to look at those 4 steps. And then we'll look at the exact enzymatic pathway after this. So step 1 is side chain cleavage. And that is acted by decimylase. Step 2 is to convert hydroxylases, or to convert hydroxyl groups into ketones. So hydroxyls to ketones. And that's done by dehydrogenases. Dehydrogenase. I am completely slaughtering this. Step 3 is the addition of hydroxyl groups, and that's done by, or that's called hydroxylation, and done by various enzymes. And the last step is to remove consecutive hydrogens, and that's reduction. So you're forming a double bond. So the steps again: side chain cleavage, dehydrogenation to form ketones, hydroxylation to add hydroxyl groups, and reduction to form double bonds. This is an overview of steroid biosynthesis. However, these are not all of the steps. This is just broken down into the most important steps, and it's also broken down so that the enzymes that are performing 2 different functions are all lined up. For example, right here we see that 17-alpha hydroxylase performs both this action and this action, whereas 3-beta hydroxysteroid dehydrogenase performs all of these actions. 17-beta hydroxysteroid dehydrogenase performs all of these actions. And I've also tried to color coordinate the circles for you as well. So, for example, these are both green, all of these are yellow. So let's take a quick look at some of these. So all of these products right here are known as mineralocorticoids. I'm just going to put mineral, but you can put O-corticoid at the end of each of these. So that's a mineralocorticoid. These 2 products right here are glucocorticoids. So glucocorticoids. Then over here we have all of the sex steroids. So sex steroids. And of course you can include the progesterones and pregnenolone as part of the sex steroids as well, although they are also important intermediates for everything else. So let's take another close look. So for example, the enzyme 21-hydroxylase and 11-beta hydroxylase, these are not present in the ovaries. Not in ovaries. And that's important because the ovaries don't need to produce mineralocorticoids or glucocorticoids. However, they do need to produce the androgens and the estrogens. So the point of this being, the exact hormone that's produced in each tissue is controlled by which enzymes are expressed in that tissue. Another example would be that 17-alpha hydroxylase. This is not expressed. It's not expressed in the zona glomerulosa. So zona glomerulosa. However, it is expressed in the zona fasciculata and the reticularis because those both need to produce the glucocorticoids. However, the zona glomerulosa only needs to produce the mineralocorticoids. Now, just to be clear, when I'm talking about the zona glomerulosa, fasciculata, reticularis, these are all different tissues of the adrenal glands. Now, steroids are mostly, so they're produced in various tissues, but they're mostly metabolized in the liver. And so you have to take into consideration liver function before administering steroids to somebody. After metabolism in the liver, they're then usually secreted through the kidneys, and so you have to take into consideration kidney function before administering certain steroids to individuals. Now, lastly, this is known as the classical pathway. So I'm just going to write classic. There are other ways for these same hormones to be produced in lesser amounts, and so those are may become clinically important when one of these various pathways are inhibited. And also, so a minimum of 17 genes or 17 enzymes are needed for the production of most of these hormones, and so of course you can see not all 17 are listed here, but this is the overview of the classic pathway. But in addition to that, each of these enzymes may be mutated in various different ways and either cause a down regulation in function or a complete absence of function. And in those, various clinical symptoms and syndromes will be present as well. We'll talk about a few in a moment. Now let's talk specifically about estrogen. Women will produce estrone, estradiol, and estriol. They produce all of them. However, they will mostly produce estradiol, a little bit of estrone, and an almost insignificant amount of estriol. So estradiol is the major secretion for a woman. Men will produce both estrone and estradiol. However, their major secretion is estrone. So if you want to kind of think of this as what is the major estrogen production for each men and women, it's easy to think, it's simpler to think of estrone as men, estradiol as women, and estriol as the placenta or baby. Now, how can we make that a mnemonic? Without sounding too sexist, which I'm not at all, if we just think of one as O-N-E, di means two, and tri means three. If you think of men as being one, women two, and the placenta or baby as being three, then you can think estrone, estradiol, estriol. However, it is still important to remember that men do produce estradiol and women do produce estrone. Estriol, even though women produce it, it's so insignificant that you can just think of it as being a product of the placenta. Now, where are these primarily formed at? So we're going to start with estrone. Estrone is usually created by conversion, so conversion of androstenedione. I'm just going to put androgens. And it usually takes place in the skin and adipose tissue. Skin and adipose. So think of a man that has a lot of body fat. He's going to start producing more estrogen than normal. And it happens by an enzyme called aromatase, or P450 AROM, P450 aromatase. Estradiol is primarily made from the ovaries. And this can happen in two different ways. It can happen directly, direct production of estradiol, or it can happen from conversion of estrone to estradiol, which brings in another important point. Estrone is also produced in the ovaries, so not in men, of course. In men, it's all conversion in the skin and adipose tissue from androstenedione. However, it can also be produced in the ovaries through directly made from androstenedione, or it can be turned into estradiol. So again, big overview. Important to remember is that androgens in the skin and fat. So I'll put skin and adipose tissue. Fat's just easier to say. In the skin and fat, they get converted to estrogen. And we'll see why this is clinically important in a second. But first, let's talk about where androgens are produced. This is specifically in females. So we have the production of testosterone. I'm just going to put test. We have androstenedione, and then DHEA, and then DHEAS, which is just a sulfated form of DHEA. DHEA stands for dehydroepiandrosterone. It's almost always abbreviated, however. So of all the testosterone in a female, 25% is going to come directly from the ovaries. 25% is going to come from the adrenal cortex, and then the other 50% is going to come from peripheral conversion of androstenedione. 50% of androstenedione comes from the ovaries, and 50% comes from the adrenal cortex. Then DHEA, 90% comes from the adrenal cortex, and 10% from the ovaries. And lastly, the sulfated form of DHEA, 100% of the sulfonization occurs in the adrenal cortex. So you've made your hormones, you've made them in all the different tissues, now they're in circulation, right? And so they're all active. Wrong. So only the free hormones are active. So free hormones are active, which is only about 2% of all the hormones that are in circulation. The rest of it is bound up to binding globulins. Most of the sex hormones are bound to sex hormone binding globulin. Some of them are bound to albumin. Now, I think that the number that I saw, and I can't find it, I haven't found it again, I think the number I saw was that like about 30%, so don't quote me on that, but just know that about 30% is bound to albumin. And what is bound to albumin is loosely bound, and it's in an equilibrium. And so of the hormone bound to albumin, it has some slight activity. However, the free hormone is what has the most activity. It has full activity, and that's about 2%. Now, when you're measuring hormones, it's important to note that it's extremely hard to just measure free hormone, and only a few labs really have the ability to do it. However, according to Williams, there is actually almost zero benefit from measuring free hormone. The most often measured free hormone is testosterone, so free testosterone. So, for example, if you were to measure total testosterone instead of free testosterone, total testosterone, if it's high, it's consistent with polycystic ovarian syndrome, regardless if you can't measure the free testosterone or not. So high free testosterone can cause polycystic ovarian syndrome, but it's almost invariably going to have a high free testosterone if the total testosterone is high. One reason for this is that testosterone decreases the production of sex hormone binding globulin, and so and then in another case, if you have normal total testosterone, but you have evidence of hypergonadism. For example, you have your the thick facial hair, you have acne being present and everything. That would be consistent. So a normal testosterone level, a high normal, which may be reducing the sex hormone binding globulin, it would still be consistent with something like hyperandrogenism. So in that one case, free testosterone may be helpful, but usually not. So I made reference to the fact that testosterone can decrease sex hormone binding globulin. And so there are things that will increase it and things that will decrease it. And I have those outlined right here. So estrogen, for example, will increase sex hormone binding globulin. Pregnancy, hyperthyroidism, both of those will increase the production of SHBG, while androgens, progesterones, growth hormone, insulin, corticoids, and increased body weight, especially central body weight, will decrease sex hormone binding globulin. So you got to keep in mind that being overweight will allow increased steroid activity. The activity is increased because the globulin that binds it and prevents it from acting is decreased. And that's another important principle, is that the amount of binding globulin is inversely associated with the activity of the steroids. So high SHBG will cause a lower activity, where low SHBG can cause a higher activity. And the significance of that is going to be dependent on how much it's increased or decreased. Really quickly, I want to talk about 3 disorders associated with steroidogenesis. One is congenital adrenal hyperplasia, postmenopausal bleeding, and hirsutism. Hirsutism is basically just saying that women are growing men-like hair in men-like places, for example, growing a beard. Congenital adrenal hyperplasia, it's an autosomal recessive disease. It's a genetic disease, and the basic biochemistry is presented here. And so what happens is you have a deficiency of either 21-hydroxylase or 11-beta-hydroxylase. Primarily, it's going to be 21-hydroxylase. And you can have anything from a mutation that causes complete inactivity all the way up to a mutation that just causes a slightly decrease in activity. So if there is a complete inactivity of these enzymes, a baby that has this disease will die shortly after birth if they don't have the right treatments initiated promptly. And what it causes, if you don't have, for example, aldosterone, you're going to have severe salt wasting. And then without cortisol, you're not going to get the negative feedback on the pituitary gland, and so your ACTH levels will probably be increased. And the increase in the adrenocorticotropin hormone levels is going to stimulate the adrenal cortex and cause the hyperplasia. Whenever you have a decrease in the activity of these enzymes, you're going to get a backup of these precursors. And with the backup of these precursors, they're going to get shifted down an alternative pathway, and so you're going to get increased endosterones. So the classic would be a complete deletion of these genes, and you need to initiate corticoid steroids. You need to start treating with that immediately. However, in a non-classic presentation of this disease with reduced activity of the enzymes, all you're going to get is maybe some decreases in aldosterone and cortisol, but also you'll have the backup of the endosterones, and it may present with pseudohermaphroditism. So that means that a female with this disease would have an XX karyotype, but she would present with more masculinized external genitalia. And then there's also a late onset version of this disease where it's the activity of these enzymes is just slightly decreased, and it doesn't become apparent until puberty, and that's when you have an increase in production of various steroids. So at puberty, you have an activation of the adrenal axis, and so you're starting to produce more of these precursors in order to get more of these adrenal hormones. But because of the activation of the precursors, this slight decrease in this activity becomes unmasked. So previously it was masked because it was only slightly decreased, and now it's become unmasked, and so you get this production of increased androgens. And that would present with, so you have a female that is undergoing puberty, and she starts to present with hirsutism. And the hirsutism basically means that the vellus hair is being transformed into a more terminal hair, like a female growing a beard. You also get increased acne as well, and we know guys go through puberty, they get acne all over their face. Women do to a slight extent, but if it's really bad, then you're going to suspect some sort of disorder with increased androgens. They may also have anovulation or decreased ovulation, and so congenital adrenal hyperplasia can mimic polycystic ovarian syndrome. And one thing that you can do to diagnose this is that you can get levels of the 17-hydroxyprogesterone. So 17-hydroxyprogesterone, you got to remember that you have increased levels of this during the luteal phase of the menstrual cycle, and so you want to try to get levels during the follicular phase. And that will prevent false positives. So earlier I mentioned that androgens can be converted to estrogens in peripheral tissue, estrogens, and that happens in adipose tissue and skin. And so a woman that has underwent menopause, she may continue to have endometrial bleeding and endometrial proliferation if she has a lot of adipose tissue. So overweight, obese women may still have symptoms of having a menstrual cycle and menses if they have a lot of body fat. Now, I mentioned hirsutism in connection with congenital adrenal hyperplasia. There's another way that you can get hirsutism. Actually, there's several ways. One important way that happens is that testosterone is converted into dihydrotestosterone, which is sort of it's a more active version of testosterone, and that happens by 5-alpha reductase. And so if that enzyme is produced in two isoforms, the first isoform is in the liver, kidney, skin, and brain. The second isoform is found in male genitalia. And so if you have an overactivity of 5-alpha reductase, then you can get a production of 5-alpha DHT. And that 5-alpha DHT can also transform vellus hair into terminal hair. And so you can treat this particular syndrome with 5-alpha reductase antagonists. There's two basic types of hormone receptors. So for the hormone to be active, it has to be present in the blood, and then there has to be a receptor that it binds to. Most endocrine disorders are either an over or underproduction of a steroid, or a hormone rather, or an over or under stimulation of a receptor. In either case, a hormone activity is either, it almost always produces a gene transcription. That's either done by transduction or by direct activation of different areas of the DNA. So transduction, we're talking about G-coupled protein receptors. So G-coupled protein receptors are going to result in transduction, and through transduction it's going to activate various genes to be transcribed and translated. So gonadotropin-releasing hormone, luteinizing hormone, and chorionic gonadotropin and FSH are all going to work through G-protein coupled receptors. So we typically know of gonadotropin-releasing hormone as being part of the hypothalamic pituitary axis. So we would expect the receptors to be in the pituitary, specifically on the gonadotrope cells of the anterior pituitary. However, recently it was also discovered that these receptors are also found on the gonads, and they're also found on the placenta. And so their exact function here is not yet completely known, but just keep in mind that in addition to the classical hypothalamic pituitary action, there's also possibly some autocrine paracrine activity with the gonadotropin releasing hormone receptors on the gonad and placenta. Now, bordering on the edge of unknown, GnRH, it's been recently discovered, has two isoforms, and the receptors have two isoforms. And so we don't really know what all of those do. So primarily we're going to only be talking about GnRH isoform type 1. So that's a 1, not an I. And we're going to be talking about this classic understanding. Now, LH and HCG both bind to the same receptor called the LHCG receptor. HCG has a higher affinity, so increased affinity. And that higher affinity, it also has a longer half-life. So it's going to have more activity on the LHCG receptors than luteinizing hormone does, but keep in mind they both bind to the exact same receptor. This is important when we talk about implantation because HCG, luteinizing hormone, keeps the corpus luteum going and acting strong, and then if there is no implantation, everything dissolves and you have the the cycle repeating itself, whereas if HCG begins to be produced, that's called rescue, and so HCG will rescue the corpus luteum from basically disintegrating into the corpus albicans and just going away, and so HCG, LH are both important in the same activity. And again, in addition to being expressed in the corpus luteum, the LHCG receptors have been found in other places: the endometrium, myometrium, fallopian tubes, the brain. Again, the activity here is still unknown. So we know what it does in the corpus luteum. Everywhere else we don't know. So we're going to talk about the corpus luteum. Now, FSH receptors, they are found in the granulosa cells, and they're found in the Sertoli cells in males in the testes, and so that is the only place these receptors have been found so far, and so that might be restricted to those areas, at least it looks like. Now, steroid hormones, on the other hand, they're going to be intracellular and sometimes intranuclear. So here you have your cell. Within your cell you have your nucleus, and the steroids will diffuse across and either bind to a receptor here or diffuse across and bind to a receptor here. Where these receptors are located, whether they're intracytoplasmic or intranuclear, depends on the specific steroid. There are two basic isoforms of the estrogen receptor, the ER alpha and the ER beta receptor, estrogen receptor alpha, estrogen receptor beta, and they're expressed differentially on different tissues. And so you can use pharmacological means to block one receptor, block one kind of receptor, while leaving the other kind alone. And so this is how various pharmacological agents, for example, tamoxifen, acts through blocking one kind of receptor but not the other kind of receptor. Also, it appears that the relative importance in each of these receptors for reproduction are slightly different. So, for example, it may be that the estrogen receptor alpha, if you don't express that, in mice it was turned off, and they accumulated cystic follicles, and they also were anovulatory. Whereas when the beta was deleted, they just had shortened or less ovulations, less menstruations. What's important to know about the progesterone receptors is that their production is stimulated by estrogen. So, for example, we talked about how estrogen, it'll diffuse into the cell, into the nucleus, it'll bind to its receptor, and then it will stimulate various gene production, gene transcription, translation. One of the genes that it looks like, or it appears to act on, is the progesterone receptors. So it increases the number of receptors of progesterone. So if you're not producing estrogen, even though you may be producing normal progesterone, it will have decreased progesterone activity. Only one androgen receptor has been identified. We're going to talk about its clinical significance in 1 second. First, I want to talk about non-genomic receptors. It appears that there may be some cell surface receptors, so cell surface receptors, for estrogen. So I'm just going to put estrogen. And so these would not have a genomic effect. They don't cause transcription translation, but they can cause other changes within the cell. They're also significant because they are going to act faster. They're going to have a faster impact on the cell than the production of proteins. And also there's been some pharmacological agents that are geared towards either inhibiting or enhancing these various receptors. So agonists and antagonists. Okay, so what happens if you have the androgen receptor and it's been mutated so that it doesn't sense the androgen's presence? This is called androgen insensitivity syndrome, and so what you could have is you'll have a male who's got a 46XY karyotype, and you'll see later when we talk about development in the last video of this, how the male and female tracts are formed, but the main thing you're going to get from this is you're going to have a male, and because he's insensitive to androgen, he's going to lack hair, so no hair, no sexual hair. And because he's a male and he produced during development anti-Mullerian peptides and factors, he's not going to have a uterus. There's going to be no uterus. which is a female part. No fallopian tube. I'm just going to put fallopians. But there will be a vaginal pouch, because the androgens are what, specifically the testosterones, are what cause during development cause the vaginal phenotype to go away and cause the penis to be formed and grow. So there will be a vaginal pouch. So phenotypically, it may look like a female at birth, and then there's going to be intra-abdominal testes. Intra-abdominal testicles, testes or balls. Now we're going to talk about this a little bit more later, but I want to prelude by saying that both FSH and LH, their production is dependent on cyclic production of GnRH. So GnRH levels will increase and then decrease, increase and then decrease, increase and decrease. And the reason for that is it prevents receptor desensitization. So that means there are two ways that you could actually stop producing high levels of FSH and LH. So you stop producing physiological levels of these two hormones. One way is that you just completely turn off GnRH production, which would be almost impossible for us to do without just destroying the hypothalamus. The other way is that you use a GnRH analog like leuprolide acetate, which will cause your pituitary to think that there's this tonic release of high levels of GnRH. So it'll look to the anterior pituitary like there's just this high tonic level. And when GnRH is produced tonically, it desensitizes the receptors, and it causes them to no longer act, and so FSH and LH will no longer be produced. So the last couple things I want to do is talk about problems with measuring hormones, and also talk about other tests that go along with hormones. So first problem is the hook effect. And so for the hook effect, let's go ahead and zoom in and talk about this specifically. So what happens if you're measuring hormones? For the most part, let's say we have this membrane, and embedded in this membrane I have various antibodies. And these antibodies, they're all antibodies for one specific hormone. So let's say that I have hormone A right here that binds to it. And it's going to bind to, depending on the level of the hormone, you're going to get it bound everywhere. So it's going to bind all of these receptors. And then typically what will happen is you'll have a second antibody that will be targeted towards the other side of that particular peptide. So there's an unbound antibody. So you have your bound antibody, it's bound into a membrane of some sort. And then you have the hormone gets added to it, and then the unbound immunoglobulin will come and bind to the top. And when it binds to the top, whenever you get both of these, the sandwich, then that causes maybe some color change, so you get blue. So if you have HCG, for example, let's say that this is HCG, then this is a pregnancy test, and so then you get the blue stripe or the pink line or whatever the pharmaceutical companies decide to make it. However, there can be a problem with this. So imagine, for example, that we have our embedded antibodies, and then we get the hormone binding to it. So we have two sides, the hormone's bound to it, but then we still have some free hormone that there was so much that we have free hormone still left over. And in that free hormone, it's going to bind to the free antibodies. And so now you don't have a sandwiching effect anymore. You have just this bound there, and then you have this bound here. Because there's no sandwich, you don't get a color change. No color change, no blue stripe, no pink cross, nothing like that, no circle. And this effect is called the hook effect. The hook effect is when you have an extremely high, so we'll say an extremely high amount of antigen.
The antigen being whatever hormone you're testing for. And whenever it's extremely high, it can bind up both all of the bound all the bound antibodies and all of the free antibodies and prevent them from binding. In fact, these will be washed away, and so you'll get no color change. Your assay is ruined. The importance of this is during some tumors that will secrete hormones, or also during different phases of hormone secretion. So, for example, at different times, HCG levels are going to be different. And so if you have an assay that will detect HCG levels almost 2 days after implantation, then it's going to detect lower HCG levels than what your body could produce, let's say, a couple of weeks or a month later. And so if you were to take the same test a couple of weeks or a month later, you may have a false negative. So this false negative, and the way in a lab that you take care of this is that you dilute your sample. So once you test, if there's a negative result, you need to dilute the sample through serial dilutions and retest each of those serial dilutions to find out if any of those present with an active antibody. And then the other thing to be aware of is that there are some non-biologically active isoforms of different antibodies, and I should have had a parentheses closed right here, or the antibody could have reduced, or the hormone could have reduced activity, or it could be non-biologically active. So the example that Williams gives is there's many prolactin, prolactin hormones. Many isoforms are not active. And so whenever you're measuring prolactin levels, you're also measuring the non-active or the non-biologically active isoforms. And so you don't know specifically if you're measuring active isoforms or non-active isoforms. And so just being aware of that and being careful of that as well. So you can do stimulation tests and suppression tests. So a stimulation test may be, for example, that you're using an HCG. You can administer HCG to test to see instead of LH. So you want to check on the various products of LH. So you administer HCG and you see if you have the correct physiological response. Another test would be the GnRH stimulation test. So you administer GnRH and then you measure FSH and LH and you see if you're getting the response that you need. So you could do this test, for example, to test to see that pubertal development is moving forward appropriately. A specific suppression test is the dexamethasone suppression test. So dexamethasone. And what this does, dexamethasone, it causes inhibition of ACTH, and without ACTH you should get a decrease in cortisol. And so if you don't have a decrease in cortisol after you administer dexamethasone, then you should suspect a primary hyperadrenalism. So primary hyperadrenalism. And that basically covers it for this part one. Next, we're going to look at how all these hormones act and interact, as well as mostly looking at the hypothalamic pituitary axis.